Done, ma'am.
Okay, Jobita, you have also joined. See, right now I'm making you people do some MCQs. So from whatever I've taught in today's, like up until now for your NEET classes from molecular basis, so those questions I've displayed on the screen. So however many you'll be able to solve, you solve it. Okay, write it in your paper and then we'll be discussing it. Okay. Is it clear, Jobita? Are you able to hear me? Yes or no?
Done, ma'am.
Done till here, students? Jobita, are you there in the class? Jason, am I audible to you? Am I audible, Jason? Students, am I audible to you all or not? Tell me.
You're audible, but the voice is very low.
Okay, right now I'm audible.
Yes, ma'am.
Okay, Jobita, am I audible to you? Done till here? Both of you switch on your videos
Yes, ma'am.
quickly. Switch on your video, students, both of you. Jobita, you also switch on your video.
Ma'am, I'm traveling right now. That's why I'm not able to turn on the video.
Okay.
Done, ma'am. Done, ma'am. Done, ma'am.
Done till here, Jason?
Done. Done, ma'am.
Oh, okay. So you're done till 16 question. Okay, so let us discuss the answers till then and then we'll move forward. Okay, so in a DNA strand, the nucleotides, they are linked together by which bond? What is your answer?
B.
Phosphodiester bond. Okay. A nucleoside, it differs from a nucleotide. Why? Because the nucleoside lacks what?
Phosphate group.
Very good. Then both deoxyribose and ribose belongs to the class of sugar called as
Pentose.
pentose. Pentose meaning how many carbons are there?
3.
5.
Oh, sorry, 5.
The fact that a purine always pairs up with a pyrimidine through hydrogen bond, what does it lead to?
Ma'am, is it C?
The uniform width, because everywhere it's going to be like hydrogen bonds, and that's the said hydrogen bond, like adenine with thymine with 2 hydrogen bonds, guanine and cytosine with 3 hydrogen bonds. It's going to be like that only throughout. Okay, the net electric charge on DNA and histone is
Negative and positive.
negative and positive. Why is histone positively charged?
Sorry, ma'am.
Why is the histone positively charged?
Um,
It is
because
positively
they're rich
charged
in
because,
amino acids, right?
yeah. So which 2 amino acids make it positive?
Lysine and
Arginine.
arginine.
Arginine. Very good. Okay. Next question. The promoter site and terminator site for transcription, they are located at which of the locations?
Uh, so is it option B?
Meaning, if you remember the transcription unit, we saw how at the 5th position, meaning 5' position, there was the promoter region, and at the 3' position, there is the terminator region. Okay, which of the following statement is the most appropriate for sickle cell anemia?
D.
Hello.
All of the above.
Can you tell me why Malay sickle cell anemia, like if a person has sickle cell anemia, there is like a very less chance that that person will have malaria. Why? Like the question is, why can't a person with sickle cell anemia have malaria?
Mhm.
So now you have studied both the chapters like molecular basis and human health. So tell me.
Mm,
It's because what happens in sickle cell anemia? In sickle cell anemia, the shape of the RBC changes from biconcave to sickle-like, and malarial pathogen, that is Plasmodium, it requires the RBC for its infection. So if the RBC has less space, where would the pathogen go? Okay, which of the following is true with respect to AUG?
All of the above.
Yes. The first genetic material could be
RNA.
RNA, right? And then DNA evolved from RNA. Okay, with regard to mature mRNA in eukaryotes, which one is correct?
B. Exons appear, but introns do not appear in mature RNA.
That's correct. And why do the introns not appear? Like, why is like intron not there? Because introns are the non-coding sequences. Can you tell me the process used to remove the introns from RNA?
From RNA?
What is the removal of introns called as?
Oh my god.
It is called as splicing.
Splicing.
Okay, the human chromosome with highest and the least number of genes are?
I'm not sure.
See, chromosome 1, it was the last chromosome to be studied, and it has the highest number of genes, and the least number is the chromosome Y. 236 or 231 genes are there. Clear?
Yes, ma'am.
Okay. Who amongst the following scientists had no contribution in the development of DNA double helix model?
D.
What did they do? Messelson and Stahl, what did they do? What was their contribution? All of the answers that you're giving are correct, but the explanations are gone. Why? Give me proper explanation also. Are you following your luck?
Ma'am,
What did Messelson and Stahl do?
ma'am, your voice is low.
Oh, I'm saying, what did Messelson and Stahl do? Like, why they do not have, like, what is the role that they played in the whole entire molecular basis?
Semi-conservative replication.
Yes, they gave the proof that DNA replication is semi-conservative. Okay, DNA is a polymer of nucleotides which are linked to each other by 3' to 5' phosphodiester bond. To prevent polymerization of nucleotides, which of the following modifications would you choose?
Can you explain this?
See, in the DNA, what happens is that every time a DNA has to be linked with another strand, like one strand has to be linked with another strand, the linkage is always 3' to 5', right? And if we talk about polymerization, polymerization is when several nucleotides join together in order to make a polymer, that is DNA. So if you have to remove that, you know that whenever a phosphodiester bond is formed, there is the, you can say, formation of a bond in between the 3' and the 5', right? Their functional groups, they form a bond. So if we remove the functional group from the second carbon, okay, or you can say the whole entire this thing, not the second, sorry, the third one, obviously, because the third one is forming the bond. So if you remove the OH group, then it will be difficult for it to form the phosphodiester bond. Clear? So this would be the answer. Understood?
Yes, ma'am.
Okay, next question. Discontinuous synthesis of DNA occurs in one strand because discontinuous, you remember Okazaki fragments?
Is it option B?
Option?
B.
Yeah, it is. Because this one had the polarity 3' to 5', and the DNA replication has to happen 5' to 3'. That is why it was made in fragments. And then later on, these fragments are joined by which enzyme? DNA ligase. It's written here only. Okay, which of the following step in transcription is catalyzed by RNA polymerase?
You're lagging.
I'm saying which of the following step in transcription is catalyzed by RNA polymerase?
D, all of the above.
Control of gene expression takes place at the level of
Transcription.
transcription. Why? Explain.
Um, it only happens in prokaryotes.
No, transcription takes place in eukaryotes and prokaryotes both.
Because they have a lack operon.
No. See, the reason why? It's because after the process of transcription only, then translation happens. So that transcript, that is mRNA, is the most important in order to form any kind of protein. That's why. Clear? Okay,
Yes,
so
ma'am.
question 17, have you done it?
No, ma'am.
Okay, now you do the question.
Ma'am, can you scroll? Done, ma'am. Done, ma'am. Done, ma'am.
Done? Done, Jaslyn? Done till here?
Yes, ma'am.
Okay, let us quickly discuss the answers. So, which was the last human chromosome to be completely sequenced?
Chromosome
Yes,
1.
chromosome number 1. Which are the following other functions of RNA?
All of the above.
All of the above. While analyzing the DNA of an organism, these many nucleotides were found. What would be the answer? Considering the Chargaff's rule?
Mm, I'm not sure. Is it single stranded DNA?
Why? Because only half of it is matching. Like the guanine and thymine are same, but the adenine and cytosine are different. In some viruses, DNA is synthesized by using RNA as a template. Such a DNA, this though you might not know because this is not there in your syllabus, but it is called as circular DNA. Okay.
Okay, ma'am.
Now, if Michelson and Stall experiment is continued for 4 generations, what would be the 4th generation ratio?
I'm not sure of this one either.
See, this one, because the normal nitrogen isotope was getting incorporated more and it was being observed more in the offspring, I mean in the next and next generation. So the highest would be for it. So that is why this would be the right answer. Clear?
Yes, ma'am.
If the sequence of nitrogenous base of the coding strand of DNA is like this much, this and the sequence of its RNA transcript would be
Option A.
option A, because RNA has uracil instead of thymine. The RNA polymerase holoenzyme is responsible for transcribing
Structural DNA and terminator region.
If the base sequence of codon in mRNA is AUG, the sequence of the tRNA base pairing would be tRNA.
Is it option B?
Option?
B.
Option
Sure.
B. C A U. Yeah, but in the opposite direction. The amino acid attaches to tRNA at its
Uh,
3'
prime
end. Yes,
end.
you remember the design? It was like that, right? Here it was the 5', here it was the 3'. This part was the amino acid binding end. To initiate translation, mRNA first binds to
Smaller ribosomal subunit.
and in E. coli, the lac operon gets switched on when
Lactose is present and it binds to repressor.
very good.